Hello everyone, and welcome to today's The Scientist webinar. My name is Nathan Nee, Associate Science Editor for The Scientist, and I will be moderating our discussion. Today, our speakers, Dr. Angela Rasmussen and Dr. Ya-Chi Ho, will discuss the positive and negative aspects of the host immune response to SARS-CoV-2 infection, and discuss how this knowledge influences therapeutic benefits. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speakers will address these during the Q&A session that follows the presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. We'll try to address as many of these questions as we can during our Q&A session. This webinar will be archived on the Scientist website, and we will send you the on-demand link via email within a couple of days. Please note that you will not be able to download the presentation slides. At this time, I would like to take an opportunity to thank our webinar sponsors. Sinobiological is an international reagent supplier and service provider specializing in recombinant antigen production and antibody development. The company has developed a comprehensive collection of SARS-CoV-2 research tools, which have been cited by 200+ papers published in Cell, Nature, Science, and so forth. Recently, within only 9 days, Sinobiological developed a recombinant RBD453F mutant, which is found in Danish and Dutch minks, to support research on SARS-CoV-2 variants. For more information, please visit sinobiological.com. 10x Genomics builds solutions to interrogate biological systems at a resolution and scale that matches the complexity of biology. Our rapidly expanding suite of products, which include instruments, consumables, and software, have enabled customers to make fundamental discoveries across multiple research areas, including cancer, immunology, and neuroscience. Selecta Inc. is a leading provider of functional genomics products and services. Its most recent portfolio additions include pseudotyped virus-based tools to study coronavirus in BSL-2 facilities and the RNA spatial profiling service on the NanoString GeoMX instrument. BPS Bioscience is a US-based original manufacturer with an innovative portfolio of over 4,000 products to assist in your drug discovery projects. As a primary manufacturer, BPS can supply products in large-scale bulk quantities and welcomes inquiries for contract and custom manufacturing within a variety of research areas. Canopy Biosciences, a Bruker company, is a multi-omics CRO providing services in both CLIA and non-CLIA environments. Core services include NanoString, RNA-seq, IHC, immunohistochemistry, and chip cytometry multiplex biomarker analysis. Canopy Biosciences offers data analysis for NanoString and RNA-seq using the Rosalind platform. Their broad portfolio appeals to a wide audience investigating the genome, proteome, and transcriptome, driving discovery and innovation in immuno-oncology research. To learn more, please visit canopybiosciences.com. Our sponsors have provided us with some helpful resources related to today's webinar, and we have posted these resources in our handout section located on the right side of your screen. You can access and download these documents at any time during the webinar. Without further ado, let me introduce our first speaker, Dr. Angela Rasmussen. Dr. Rasmussen is a virologist and affiliate at the Georgetown Center for Global Health Science and Security, who studies host responses to infection by combining classical virology with modern systems biology approaches. Her research objectives are to identify host response signatures predictive of infection severity or disease outcome, and host pathways to target drug development or repurposing. Dr. Rasmussen is particularly interested in viruses that are highly pathogenic and newly emergent or likely to emerge because of climate change, land development, or ecological disruption. Dr. Rasmussen has employed collaborative cross-CC mouse models, which provide an expanded range of disease presentations to study viral characteristics. She developed a CC mouse model of Ebola virus disease utilizing the diversity of CC mouse disease phenotypes to study genetic and transcriptomic factors underlying disease severity in humans. She is currently evaluating CC mouse models to investigate sex-specific host responses to viral infection, as well as disease presentation caused by other viruses that pose a major threat to global public health, such as dengue and influenza viruses. Ultimately, these host response profiles can be used for translational or biodefense applications, such as diagnosing infection, predicting disease severity, informing vaccine design, and developing or repurposing host-targeted drugs to impair virus replication or reverse pathology. Before we start, let's make sure your slides are up and running and everything looks good. So please take it away, Dr. Rasmussen.
Well, thank you so much, Nathan. I'm just going to go right into my presentation. So before I start all of my presentations, I like to give a territorial acknowledgement and equity statement. And I'm presenting today from Seattle. That's the unceded ancestral homelands of the Duwamish people. And much of the work presented today was performed on the lands of the Duwamish and the Lenape people of Manhattan Island. I acknowledge and honor the first people of these territories, as well as their tribal governments, their histories and ancestry, and their roles today in caring for these lands. And I'd also like to acknowledge that there's a history of systemic inequity in academic science that has spanned for decades, and indeed for centuries. The institution where much of this work was conducted, Columbia University, was founded using profits from the transatlantic slave trade and excluded women and people of color from the academic community for nearly 2 centuries. This has left a long and painful legacy of racial and gender-based inequality that continues to this day. I would encourage all of the folks attending this webinar today to consider how you can contribute to making biomedical and public health research a more equitable enterprise. So I'm going to begin by just giving a brief introduction to SARS-CoV-2, which is the virus that causes COVID-19. This virus, even though it has really profoundly impacted all of our lives around the world, is actually a pretty standard beta coronavirus. It is this small particle that has a genome of about 30 kilobases. It's a positive sense, single-stranded, non-segmented RNA virus that's in the family Coronaviridae, and it is classified or taxonomically grouped with the beta coronaviruses within its own subgenus, the sarbecoviruses, or the SARS-like coronaviruses. SARS-CoV-2 has unique evolutionary origin and host adaptation, and this impacts how it will interact with the immune system of a given host. We don't actually know that much about the origin of this virus, but its progenitor viruses likely evolved in bats. It's unknown whether there was another intermediate species that the virus circulated in before becoming adapted to humans, but the virus that is causing the current pandemic is SARS-CoV-2, which is adapted well enough to human hosts. It also may be adapting to other new hosts, which have the potential to transmit back and forth between the human population anthropodically and zoonotically, including cats and mustelids, such as minks that have been recently implicated with large outbreaks on mink farms in the Netherlands and in Denmark. And its mechanisms for adaptation, because it is a non-segmented virus, are through mutation or recombination. And this does become important when considering immune responses over the long term. Since this virus can mutate, as all RNA viruses can, that may have implications for immunity down the road. So viruses do evolve as they spread through a population, and this is the current next strain data showing some of the mutations that have been acquired from different isolates all around the world. And while this virus does have a lower mutation rate than many other RNA viruses due to the action of an exonuclease that confers partial proofreading capabilities, this virus is, like every other RNA virus, again, evolving as it spreads through the human population. And we don't know that much about specifically what this evolution means in terms of the immune response. One of the best characterized variants, this so-called D614G mutation in the spike protein, has been shown to change the virus's properties a little bit. It increases fitness or replication in the upper airway of Syrian hamsters, which is becoming a leading model for studying COVID-19 pathogenesis. It also appears to be transmitted more efficiently in hamsters, but importantly, it doesn't change sensitivity to neutralizing antibodies. So this is something that is still very much an active area of research, trying to understand how these changes in the viral genome might impact the way that the virus interacts with its host, and consequently the implications for immunity or COVID-19 pathogenicity. So here's some host virus interactions that can impact immunity, and this is just kind of an overall scheme of the entire immune response. And pictured here is influenza virus, but it's a similar situation for SARS coronavirus 2 in that it infects ciliated cells in the airway. But before it even gets there, there are passive immune mechanisms intended to keep the virus from infecting cells. And that are things like mucus and collectins. These are proteins that will just really bind a virus and keep it from doing anything, keep it from getting to the cells that it could actually infect. But if you're exposed to a high enough dose of the virus, then it probably will encounter cells that do express the receptor, in this case, the molecule ACE2. It will enter those cells, begin replicating, and then a whole bunch of different types of immune responses are triggered. Initially, the innate immune response is mediated both by specialized innate immune cells as well as various pathways within an infected cell. This type of response is rapid and immediate, and it's also nonspecific, but I put an asterisk next to that because it's sort of nonspecific. Some of these cells, like NKT cells, for example, really kind of bridge the innate and adaptive immune system. And in addition to that, one of the main mechanisms for recognizing viral infection are through so-called pattern recognition receptors, or PRRs. This includes things like the toll-like receptors pictured here, as well as the Rig-I-like receptors, which are cytoplasmic pattern recognition receptors. And they recognize specific motifs that may not be specific to an individual virus, but are generally associated with viral replication, such as double-stranded RNA. So it is nonspecific, but it's not completely nonspecific. Within 1 to 2 weeks, the adaptive immune system takes over, and this is a very highly specific immune response. This is specific for the antigen that the virus has in terms of its viral proteins. So this is specific to SARS coronavirus 2 and not other coronaviruses or other types of viruses. And this involves B cells, which produce antibodies, as well as T cells, which come in 2 basic flavors. There are CD4 helper T cells that coordinate immune responses across the adaptive and innate immune system, as well as CD8 positive killer T cells, which go out and kill infected cells, as is shown here. They're also called cytotoxic T lymphocytes, or CTLs. This is what we think of often when people talk about the immune response. They think of antibody titers, but the immune system, as you can see here, is actually much more complicated than that. So today I'm mostly going to focus on the innate immune system and how that interacts with the adaptive immune system, and the implications for long-term immunity that innate immune responses contribute to. So this is an overall diagram that's very simplified, but it shows the interferon system. And interferon is a cytokine that was so named because it was observed that it interfered with viral replication. So type 1 interferons in particular are really critical molecules for the innate antiviral response. When a virus gets into a cell, it comes into contact with one of these pattern recognition receptors. In this case, TLR3 or TLR789 are pictured here. They are linked to endosomes, which are compartments that the viruses are internalized in. In addition to that, there is Rig-I and MDA5, which are in the cytoplasm. And once the virus has completed fusion and put its genome into the cytoplasm and begins replicating, these can recognize double-stranded RNA motifs that are associated with viral genome replication. Both of these pathways end up turning on interferon signaling. They result in the production of interferon, which then is secreted from the cell, binds its receptor on another cell, and activates this JAK-STAT signaling pathway, which then results in the expression of so-called interferon-stimulated genes, or ISGs. And I included this separate picture of these Spartan soldiers here as the ISGs, because ISGs come in a variety of different flavors and functions. They do all sorts of different things, from directly attacking viruses or viral proteins or viral replication products, to turning off or modulating the host to deter or suppress viral replication. So there's a lot of different specialized ISGs that have different functions, but all of these processes are really critical to mounting an effective antiviral response, particularly early in the infection. So viruses have evolved over time, all these different mechanisms for blocking this system. Viruses can block pattern recognition by interfering with these pattern recognition receptors' ability to recognize virus motifs. They can silence signal transductions, so they can dephosphorylate some of these signaling molecules, prevent them from basically getting into the nucleus and starting to express interferon itself. They can shut down interferon production or block its secretion from an infected cell. In addition to that, in the cell that would be the target of that interferon signaling, they can block the JAK-STAT signaling pathway by a variety of different mechanisms, one of which is blocking STAT dimerization. So activated STAT molecules have to form these dimers before they can enter the nucleus and act as transcriptional activators. It can block nuclear translocation. And it can also prevent ISG expression. And some viruses have also evolved the ability to inhibit specific ISGs that are important for counteracting those specific types of viruses. So the virus and the host interactions and the immune response is really a very complicated dance in which either partner is constantly trying to step on the other partner's toes or mess up their steps. And it's, an ongoing struggle throughout evolutionary history. That's a really fascinating area of study from my personal opinion. So how does this impact SARS coronavirus 2 and COVID? Well, some work out of Ben Tenover's lab at Mount Sinai shows that SARS coronavirus 2 infection in ferrets and in cells, as well as in human patients, really profoundly suppresses both type 1 and type 3 interferon responses. And type 3 interferon responses are also thought to be important for innate antiviral function. They do that more profoundly than other common respiratory viruses, such as seasonal influenza virus. And one consequence of this is that in shutting down these interferon pathways, they're actually also shutting down regulation of many of the other inflammatory pathways that talk a lot. There's a lot of crosstalk and redundancy between many of these antiviral signaling pathways, and that does include signaling for a number of different pro-inflammatory pathways. So the consequence of shutting down type 1 interferon is not just that the virus can grow better, but also that it upregulates the expression of all these inflammatory cytokines and chemokines that then recruit inflammatory cells to the site of infection in the lungs. And this has been proposed as a mechanism for which COVID-19 can actually occur outside of SARS coronavirus 2 infection. So SARS coronavirus 2 infection, is shown by the same group, does induce very unique host responses that are very different from the types of responses that are induced either by influenza A virus, and influenza A virus that is unable to antagonize interferon, or treatment with interferon itself. The gene expression profiles of cells infected with SARS coronavirus 2 are very different, as shown here on this principal component analysis. When you look at the pathways that are induced, like what do these genes actually do biologically, you can see that they also have very significant differences in terms of the genes that are classified as being involved in interferon signaling or response to viruses, response to cytokines, and regulation of MAP kinases, which is an important pathway for inflammation. So it really does look like SARS-2 is able to manipulate the host response in a way that is probably favorable to its replication. And as I mentioned just a moment ago, the effect of innate immunity on inflammation is really important in terms of the pathogenesis of COVID-19. So forgive me, this diagram is a little confusing, but what goes on here, this is basically showing two different scenarios. It's showing the virus infecting a cell and the effects that it has on that cell directly. And then it's also showing some of the responses that happen. So all these inflammatory cytokines and chemokines start getting secreted. There's all this communication and positive feedback with both cells of the adaptive immune system as well as cells of the innate immune system, such as monocytes and macrophages. Then it can go one of two ways. You can have a healthy immune response in which inflammation is controlled effectively. Antibodies are produced, T cells are produced and begin eliminating infected cells. Virus release is stopped, replication is stopped, and the infection is cleared. There's this other situation, however, where there's a dysfunctional immune response in which all these pro-inflammatory cytokines begin being secreted. People are calling that the cytokine storm. It basically means uncontrolled expression of pro-inflammatory cytokines. This can have a pretty profound impact on the function of T cells and B cells down the road. So it is thought that an aberrant immune response to infection really leads to severe COVID-19. And that's what I'm going to talk about a little bit more today. So we don't really know much about the mechanism by which SARS coronavirus 2 can antagonize the interferon system. But one group has actually started to look at this fairly systematically, and they've identified a variety of different viral proteins that have an effect on these pathways that I just told you about. So they can both impact this in terms of shutting down signal transduction from the pattern recognition receptors. In this case, they're showing RIG-I, and that prevents interferon from being expressed at all. In cells that are not infected, cells that are infected but have not yet had interferon be activated, they can also impair STAT signaling. So this is involved in signaling and expression of those ISGs that I mentioned. So they can block that in a couple of different ways, both by blocking the formation of STAT1 and 2 activated dimers, as well as blocking the ability of those activated STAT1 and 2 complexes from entering the nucleus and beginning to express ISGs. So we wanted to know, my group wanted to know, and I'm just going to show you a little bit of primary data, how this worked systemically, because we know from Ben Tenover's work that SARS-2 can suppress interferon, but what is the consequence in a model of actual COVID-19 disease? So we used the rhesus macaque model, collected whole blood samples from infected rhesus macaques over a 21-day time course, and then we performed RNA-seq to identify systemic host response signatures in the peripheral blood. And it's important to note that the rhesus macaque model is a model of moderate COVID-19. These animals do have pathological evidence of disease, as well as evidence of pneumonia and infiltration on X-ray. However, they don't really get that sick and they recover. So this is not a model of really severe COVID-19, but it is a tractable animal model to study this in. So what we saw when we looked at our data was actually very different from what the Tenover lab saw in infected lungs. And it's important to note also that in the peripheral blood in these animals, there was absolutely no virus. So we're looking at cells that are not infected, again, systemic responses. And we saw that interferon, all of these pathways related to those innate antiviral responses that I just told you about, are actually really upregulated early on in infection, suggesting that while it may be suppressed in the lungs, the cells that are actually being infected, systemically, there is an effective immune response. And we also saw, looking at the cellular level, this is a method of using transcriptomic data to infer which kind of immune cells are driving those signatures. We saw that there was an increase in relative myeloid and phagocytic cells. So these cells that are involved in the innate immune response and are really important in controlling inflammation. So antigen presenting cells that go on to activate T cells, as well as macrophages that are really important inflammatory effectors. Those are increased in the peripheral blood, which makes sense with what we know in the context of an acute infection. These cells are being mobilized from the periphery to go into the lung and begin clearing the virus infection. We also see an increase in plasmacytoid dendritic cells, which are highly specialized to secrete type 1 interferons, suggesting that they are mounting effective systemic interferon responses. And in this model, as I mentioned, this is moderate COVID-19, but these systemic innate immune responses appear to be intact, even if they're not in the cells that are directly infected. This seems to be supported by some data that came out of the clinic, in which decreased interferon and ISGs were observed only in the peripheral blood of patients who had severe or critical COVID-19, but not in cases of mild or moderate COVID-19, which is consistent with what we see in the rhesus macaque model. So that led to us developing a hypothesis that we're trying to test now, that while direct infection may suppress interferon and result in this increased inflammation, leading to this cascade of aberrant immune responses, including aberrant adaptive immune responses. In mild to moderate disease, if it's controlled in the periphery, if you have these early interferon responses, you can control inflammation so that it's only directed at clearing the virus, but not causing the harmful cytokine storms that are associated with severe disease. In severe cases, it's likely that these systemic responses would also be suppressed, leading to that uncontrolled inflammation. But as I mentioned, we still need to do a lot of work to test this hypothesis. And I'm just going to conclude here with some implications for adaptive immunity. So as I mentioned, with SARS coronavirus 2 infection, the innate immune response comes up first. That results in the activation of a number of different kinds of cells, but especially dendritic cells, which are really important in activating T cells and initiating these adaptive immune responses. That leads to cytokine production that regulates T cell polarization. For example, in Th1 polarization is thought to be really important for mounting effective antiviral responses in the adaptive immune system. Also induces a class of T cells that are important for regulating inflammation, as well as T follicular helper cells that are involved in mediating antibody responses and also immunological memory. And this is, you may have heard that some patients do have declining levels of antibodies in the months shortly after they've recovered from COVID. And many people have pointed to this as waning immunity, but I don't think that's really a correct interpretation of it. I think that it's very normal for high levels of IgG and neutralizing antibody to be produced right during the end of and directly after an acute viral infection, but then those antibody levels normally drop down and to a baseline level, and sometimes they can be undetectable, but that doesn't mean you don't have immune memory. However, we have also observed that there are some cases of reinfection, and while it's very unclear whether those people lost immune protection or never had it to begin with, it does suggest that in some people anyways, they may not be developing as robust of an immune memory response. And that was really bolstered by this study that looked at germinal centers or places basically where helper T cells, T follicular helper T cells, are activating B cells to produce antibodies, to make those antibodies better and inducing immunological memory. In patients who had died of COVID-19, they didn't have observable germinal centers in their spleens or lymph nodes where these normally form. And they were also associated with these cells that were macrophages that were secreting a lot of TNF alpha, which is a really potent pro-inflammatory cytokine. So that may have actually led to a block in T cell differentiation, leading to a loss of immune memory. And I think of this as somewhat of a snowball effect. Loss of immune memory is a critical problem, especially if we're talking about, how well protected are people who've had COVID-19. But you could trace that all the way back to their failure to mount a functional innate antiviral response in terms of interferon expression. So this is something, again, that does require a lot more investigation, but I think it's a really interesting topic, and I hope that this segues nicely into Dr. Ho's presentation, which I think is going to focus more on the adaptive immune system. So I'm just going to end there by acknowledging the collaborators who contributed to that work that I showed you from my lab, and I'm happy to take any questions during the Q&A session. Thank you.
Thank you, Dr. Rasmussen, for that wonderful presentation. As a reminder to our audience, you may ask questions at any time using the Q&A tab. Our panel will have a chance to address these after our next speaker. Our next speaker is Dr. Ya-Chi Ho. Ya-Chi Ho is an infectious disease physician at the Yale School of Medicine who studies virus-induced immune dysfunction at the single-cell level. She combines virology, molecular biology, immunology, and single-cell RNA sequencing to study HIV host interactions in CD4 positive T cells. Her ultimate goal is to identify mechanisms of HIV persistence and to develop HIV cure strategies. Dr. Ho received her MD in 2002 and did her internal medicine residency from 2002 to 2005 and infectious disease training from 2005 to 2007 at National Taiwan University Hospital. She became interested in virology research after taking care of HIV-infected individuals and SARS-CoV-1 infected patients in 2003. After 1 year of practice as an infectious disease attending physician in Taiwan, Dr. Ho focused on research. She received her PhD at Johns Hopkins University in 2013 and continued on her postdoctoral work with Robert F. Siciliano. Dr. Ho started her lab at Yale University School of Medicine in 2017 as an assistant professor in microbial pathogenesis and medicine. She is a Yale Top Scholar, Gilead HIV Research Scholar, and an investigator in the AIDS Clinical Trial Group. So let's make sure your slides are up and running and everything looks good. So please take it away, Dr. Ho.
Thank you for having me. And thank you so much, Angela, for such a wonderful talk. I really learned a lot from that. I'm an HIV researcher using single-cell methods to study the rare and heterogeneous population of fatal reservoir and HIV-induced immune dysfunction. So today I'm going to talk about how single-cell methods that we learn from studying HIV can provide detailed and powerful immune profiling in COVID-19, focusing on the role of CD4 T helper cells in COVID-19. So why is COVID-19 different from other viral respiratory infections? We actually have learned a lot in SARS-CoV-1 from researchers such as Stanley Perlman and Ralph Barrick and many others. So for example, in SARS-CoV-1, in the animal model developed by Stanley Perlman on the left, they've been showing that, well, there is already an interferon dysregulation that's being shown in this animal model. So early studies show that typically during viral infections such as influenza, interferon should kick in and stop the replication of the virus, as Angela just talked about. However, in this model, they show that because SARS, both COVID-1 and COVID-2 now we know they have ways to block interferon responses, then this interferon response kicking a little bit too little and too late. Because the timing is too late, so then the bridge between innate and adaptive immunity becomes messed up. So then instead of clearing this viral infection, then we see a lot of like cytokine storms that dysregulated adaptive immunity in SARS-CoV-1. The second difference is that unlike the common cold coronaviruses that only colonize the upper airways, and unlike SARS-CoV-2, the SARS-CoV-1 that's infecting the lung, but not much in upper airways, so that its spreading is not as efficient. SARS-CoV-2 is both efficient in spreading because it's in the upper airway in the nose, and it's also like damaging the lung terribly. So we have a lot of transmission, a lot of mortality, and that is different from the previous SARS-CoV-1. So we want to understand CD4 T cells because we want to control inflammation and disease severity. Very frequently, when patients visit the hospital, they presented, say, with some cough and some kind of fever or dyspnea. They're usually after 5 to 7 days of the real viral infection, or even longer, like, after 5-7 days of symptom onset, they visit the hospital. But if you think about it, at this time when they visit the hospital, their viral load already peaked. That it's already that dysregulated interferon response and cytokine storms that is kicking in. So at this time, it may be too late for them to receive an antiviral therapy. It's just too late. So to help these people and reduce disease severity, we need to understand what happened in this inflammation, because many patients, when they presented to the hospital, that what we're fighting against is inflammation, and it's already too late to stop viral replication at that time. You can still use antivirals, but a lot of things damage are done by inflammation. And second thing is that we want to understand how to build long-lasting and protective immunity. This is reviewed by Stanley Perlman. What is shown is that we don't develop much common cold coronavirus antibodies. They just wane and you get a repeat infection. It's shown in previous Nature paper in September. But so SARS-1, at least we know, seems that there is some kind of long-lasting immunity that can be detected up to, say 11 or 7 years in the follow-up. We still don't know about what happened in SARS-CoV-2, and we're trying to understand this. So why do we care about CD4 T cells? I would like to remind the audience that there is another webinar that's freely online available talking about B cell responses and antibody durability that is discussed separately. That if you're interested in that, you can feel free to go to that webinar link. But today we're going to focus on CD4 T cells. We care about CD4 T cells because they're the central orchestrator of adaptive immunity. So basically, CD4 T cells have 3 jobs. One, it induces robust CD8 T cell responses. So it's a CD4 T cell help for a CD8 T cell. So CD8 T cells are the main effector, the main army to kill infected cells. So it needs CD4 help to induce this robust response. And second, CD4 T cells themselves can kill infected cells through direct cytotoxicity or through effector cytokines such as interferon alpha, interferon gamma, and TNF alpha. So they can have a direct killing. Especially in CD4 T cells, there are a small subset of them. They're antigen-specific CD4 T cells, for example, in the case of SARS, that these are the cells that respond to SARS antigen stimulation, and they will become the memory that later, when we get exposed to this virus again, it's these antigen-specific CD4 T cells that will maintain the memory response. The third job that CD4 T cells do is that these are special T follicular helper cells that they live in the germinal centers and they help B cells. So basically, B cells make the first version of antibody. They don't really work very well, but with the help from T follicular helper cells, they're going to go through several rounds of affinity maturation and class switching so they make good, potent, and neutralizing antibodies. So we think CD4 T cells are very important in many viral infections, including HIV and COVID-19. And the second observation is that why is CD4 important? Is that very early as like January, people already noticed that in severe COVID-19 patients, that CD4 T cell count are like largely depleted. This is about 200 per microliter. It's like we will almost call this as AIDS if it's an untreated HIV infection, but this is not AIDS. But we do see like a CD4, like low CD4 count in severe patients across multiple studies. And not just that their count is low, but it seems like they may have some impaired function, such as interferon gamma expression. So we want to understand what is wrong. And the third thing is that we learned from previous lessons in SARS-CoV-1. Back then in SARS-CoV-1, that this is a Ralph Baric study that, we want to develop SARS-CoV-1 vaccine. The easiest way to do it is to take the virus and inactivate them, add an aluminum adjuvant, inject into mice. This is how, say, influenza vaccine is given. So it is great that it does induce a robust antibody titer and good response. However, something went wrong, that if you just use this a very typical traditional way to build a vaccine, somehow it induces Th2 cytokines such as IL-5 and IL-13. So instead of the viral killing Th1 cytokines, it's toward Th2 phenotypes. And what is even worse is that it brings a whole lot of eosinophils to the lung. So then aged mice, like older mice in this experiment who got the vaccine, and when you challenge them with a real infection, they actually perform worse because their lungs are stuck with these eosinophils and they have worse outcome, worse mortality. So basically the vaccine just get it worse. So we shouldn't replicate this without knowing why. But this is showing the importance of whether we can make a vaccine to direct toward Th1 responses and how do we avoid these side effects. So, but later on, we know this is because of aluminum adjuvant, but we just need to understand that CD4 T cells are also important in vaccine development. So why do we see a lot of papers having like single cell, high dimensional profiling, a whole lot of those? And is that just because they are new technologies, they're just fancy? So turn out CD4 T cells or immune cells are not really just one single cell, like one single type of CD4s. So just CD4s, they can have different polarization, they can function, and cytokine profiles, exhaustion, activation, memory phenotype, antigen specificity, many different things. So taking a regraph example that if we look at all the cells all together, it's trying to guess out what this glass of smoothie has. We can have many different kinds of fruit. We just don't understand how many fruit and which component that is. But if we look at them at a single cell level, then you can see the proportion of each fruit. You can name them, you can call them, and you can look at the different, for example, functions or transcriptome in individual cells. So this helps us to understand the rare, heterogeneous, and unknown populations of immune cells. And that's why we commonly see single cell technologies in these COVID-19 papers. So in the very beginning, we're trying to keep updated on this current research and with many great papers from many like outstanding researchers around the world. And those are just very helpful and informative. The problem is that we notice that there's not only an outbreak of virus, there's also an outbreak of papers. It's just quite hard to keep up with them. That when we write, there's commonly like 4 Lancet papers in 2 days, for example. It's just very hard to chase after them. So we're just sharing how we look at these papers when they come out. So we notice that first, disease severity matters. That whenever we look at a paper, is it done in mild cases or in like postmortem analysis, it's in mild or severe. And second, the timing of the collection of the sample is important. For example, if the samples are collected right at symptom onset, we can recapitulate something viral replication going on. But whenever the blood is taken during ICU admission, usually it means that there's a lot of inflammation going on. And their samples taken like during convalescence, like convalescence plasma, or during recovery phase after they're discharged, they may have robust CD4 T cell responses, but they may not recapitulate what's ramping up in the very beginning. And of course, age and sex matters. So we just look at these different points when we like navigate through the literature. So where's the battlefield? Of course, the lung is the battlefield of COVID-19 because like respiratory failure is the major reason that this is causing death. But to study lung is not very easy because these patients already have a very fragile lung. You cannot just take a biopsy, poke a hole, and the patient may just die because of this too invasive procedure. So the researchers would take bronchial alveolar lavage fluid. Basically, you just throw some saline in and wash them out and look at what's the cellular component in these. And then, so there's single cell landscape analysis of these immune cells in the lung. They found that, so the key question is who is making the cytokines and who is causing the cytokine storm? There's been argument like who's doing it. It seems that in this paper, it's shown that it's the macrophages in the lung. They're making IL-6, and F and a whole lot of chemokines. So then it's the lung macrophages that is initially making all these cytokines that is likely contributing to the overall systemic inflammation or cytokine storm. They do find expanded clones of cytotoxic T lymphocytes, such as CD8 T cells in them, but it seems like they're responding to this infection secondarily. For example, in moderate patients, they have increased T cell clones for proliferation. But in severe cases, these proliferation is gone, suggesting that the T cells want to come and contribute and control the infection and proliferate. But in severe cases, these T cells are probably not sufficiently proliferating to maintain them. Another study showed that, they look into different lungs lavage samples across different disease severity. So what they found is that these are the cytokine profiles from mild to moderate to like the severe disease. And you can see that these bubbles are looking like bigger and brighter and more. This is because the cytokine profile seems to be more like in those people with severe disease. So what this paper found is that it's the infected lung epithelial cells, they have innate sensing. They sense out there's a virus coming on. They started to scream out like cytokines. Then they recruit non-resident macrophages, which means that they secrete these chemokines, and macrophages in response to these calls like flush like into the lung. But supposedly this should contain the infection. They're coming to help. But because of the cytokine dysregulation, these macrophages come and sit there and secrete a whole lot of chemokines, and making exudates. So then the lung should be like a thin layer of balloon having lots of air exchanging, but with all these immune cells squeezed into the lung with exudates and fluid, that people feel that I just cannot breathe because my little balloon is now like a water balloon. You cannot breathe, and that's called respiratory failure. But this is showing that non-resident macrophages is a culprit producing cytokine storm. So what can we do? So this recent paper published this week showed that there are ways to intervene into this cytokine storm. For example, in this mouse model, they use TNF alpha interferon gamma antibodies to block, hopefully, this cytokine storm in the very beginning. So then block the downstream signaling that Angela just talked about. And they show that this can decrease motility and decrease tissue damage. Whether this can be used in humans is still unknown, but it's a proof of concept mechanistic analysis of what we know about what's being dysregulated and how we can propose a treatment or immunotherapy. The second battlefield is the lymphoid tissue. You're like, why? So lymphoid tissue is the home of lymphocytes, and also it is the home of both T cells and B cells. And this is where B cells getting their education and make high affinity antibodies. On the left, this is a normal lymph node. You see these like bubbles. These are the germinal centers or follicles. So basically, there are T cells and B cells like interacting in there. So then B cells make antibody and go to T follicular helper cells. And then T follicular helper cells will help class switching and help affinity maturation. And to the end, it makes a good and potent and neutralizing antibodies. But this is the same study that Angela talked about before, that in diseased patients, when they look at their lymph node, it's just all wrong. So on the left, it's like a typical elementary school. You have the structure of a school. You have teacher called T follicular helpers, and you have B cells called students. But in COVID-19 cases, it's all messed up, that the structure is not there. And so this means that there's no way that B cells can go through these guided maturity of affinity maturation, so to make better and better like antibodies. It's just all wrong. Like the structure is not there, and the T cell is lost. So this paper concluded that because the loss of T cell follicles and may be a reason why some patients may not develop durable humoral immune responses in COVID-19. So if you don't have T cells, you may have problems with your antibody production. And finally is the blood. So we take blood because you can it's less invasive. You can follow patients longitudinally, and you can look at the immune system using this window. We don't mean that blood, like immune profiling, is the same as lung, because they cells do different things at different sites. But this is a window for us to understand the immune system. Especially in the blood, we're going to talk about two things. So first is CD4 T cells generally overall. The other is the antigen specific, like SARS-CoV-2-specific CD4 T cells. That is the one that's going to develop memory. So what happened to them? There are typical ways to look at CD4 T cells in the single cell level. So one is using a protein-based method, such as flow cytometry or mass cytometry. So this is CyTOF-based, or this is VAX-based. So basically, you have a selection of antibodies, say from 40 to up to 200. If you do CytSeq, for example, that you have the antibodies, known proteins that you want to look at. You can stain them and look at them. And second is the RNA-seq-based method, that you use RNA-seq, so it's a unbiased genome-wise profiling of the transcriptome of individual cells. The good thing is that you don't need to pick what's already known. You just let the data tell you what's up and down. The bad thing is that RNA transcription doesn't really reflect a protein expression. So if you join them together, plus clinical information, plus a viral load, plus plasma interferon cytokine levels, integrating all these immune data all together, then we'll reach so-called high-dimensional immune profiling. For example, this one is John Hueri and Michael Beck's study looking at deep immune profiling of COVID-19 patients. Then combining all these data, high-dimensional flow cytometry of immune cell markers, and then with clinical data and cytokine levels, they found that this COVID-19 response it's just very complicated. There are different people who may respond differently. They may not be like a one shoes fitting all immune response, but for example, there is something that's going to come. For example, some group of people having a highly activated CD4 and CD8 T cells that's also seen in other studies. This is Akiko Iwasaki's analysis looking at COVID-19 patients from very mild to very severe across different time points. So this is a Yale impact study, a prospectively archived, like planned and archived longitudinal study to look at immune profiling over time. So what she found is that, yeah, this is reminding us the Th2 dysregulation in the SARS-CoV-1 vaccine study. Remember that we say that in that vaccine study, IL-5 and IL-13 was upregulated. And same as here, that Akiko's group found that there is an upregulation and misfiring of Th2 phenotype of IL-5 and IL-13, that SARS infection should induce type 1 Th1 to fight against the virus, but there is this Th2 misfiring. We still don't know why, but this is interesting. And second is that there is an upregulation of interferon alpha and interferon gamma, like interferon alpha. It's a little bit too late, similar to what we learned in SARS-CoV-1. It's there, but severe patient, you see this peaking up a little bit late. The moderate patient, it's like coming down like early. So this interferon level is correlating with viral load in the nose, and then it's correlating with the severity, that the more severe you have a stronger and higher level of interferon response. Then using other ways to look at this, for example, if you look at this another study using systemic biology, there are several like different like mass cytometry and site-seq and different studies all combined in this study. You can see that in ICU admitted patients in this blue dot, it seems like their interferon are peaking a little bit late. And those moderate patients in the purple dots, they're somewhat peaking early. Well, we're not sure, but this is somewhat similar to what Akiko's group has found. What this paper found is that interferon response is a little bit too little and transient. It seems to be not enough. But what is found is that if you look at the plasma level of interferon, it's a protein floating in the blood. This can affect the transcriptome of cells, that the cytokine in the blood is affecting the transcriptome of cells in the blood. So it's correlating with each other. So your immune dysregulation in the blood is affecting the immune cells and in their behavior. So what this group found was that CD4 T cells have increased expression of inflammation-related genes, and like they're just more activated, and they have these like FOS, June, DUS, and these are likely the things that they propose to cause like dysregulation of CD4 T cells, and they have lower anti-inflammatory genes in COVID-19 patients. This is a study done by Yale showing that there is a dyssynchrony between the innate and adaptive immune system, that it's just dysregulated because the sequence of bridge is just sort of broken. So finally, here's one question that we asked in the very beginning. So why do COVID-19 patients have low CD4 T cell count? Like HIV infects CD4s, but SARS don't. SARS virus infection doesn't infect like CD4 T cells. So why did they die and where did they go? So by single-cell RNA-seq, they found that by comparing COVID-19 and influenza infection, they found that there is a signature that is different in COVID-19. You see these like IL-10, IL-6, TNF, interferon, that these things are going wrong. And then when they compared between COVID-19 and control of the donors, they found that, it is TNF, like transcaspase Fas, that is it's these ones that are upregulated, so that it is contributing to the death of CD4 T cells. So that's why they may have a lower CD4 T cell count. So the last study I'm going to talk about in the bulk CD4 level is that this study found is that if you look at viral load in the blood, interferon alpha level in the blood, and CD4 T cells, you can sort of figure out one story, that because SARS interfere with interferon signaling, you have lower levels of interferon alpha in critical patients. Because of that, then your SARS infection cannot be controlled. So you have higher level of SARS viremia systemically in these patients. Then because innate immunity cannot control this viral infection, you're going to call for the help for adaptive immunity. But this is just too much overwhelming activation. So you see activated CD4 T cells expressing exhaustion phenotype. So that's how I would link like the SARS causing innate immunity dysfunction and then causing adaptive immunity failure. So what happened to SARS-CoV-2 specific CD4 T cells? So are they functional? So to look at them, the standard way to look at SARS-specific or antigen-specific CD4 T cells, either you need to have the HLA restricted like tetramer, and it's going to be hard to design for each individual, or you use interferon gamma like cytokine secretion, but those it's kind of complicated because you need to fix the cells to look at intracellular cytokine staining, but then after that you cannot do RNA-seq because fixed cells have very poor quality. Anyways, so Shane Crotty's group developed this activation-induced marker staining like back in 2016. Basically, if you have CD4 T cells in the presence of APC or just PBMC, you throw into these peptides, so it's like 11 to 15, like amino acid, kind of little cut out like peptide pools. Then if they're antigen specific CD4 T cells, they'll respond to this stimulation just over like 9 to 24 hours of stimulation. So if you just sort out cells that are expressing activation markers such as CD69 or the signal 2 of T cell receptor signaling such as CD137 or CD154, if you just find these cells that they express these activation induced markers in flow cytometry, then you can sort them out, count them, and answer what's going on with SARS specific CD4 T cells. So first, people found that with severe patients, you have more CD4 T cells, that you have more SARS specific CD4 T cells. So they're there. Not only they're there, they're functional, that they proliferate upon stimulation and they express interferon gamma. So through this detailed single cell analysis, now we know that they do have robust T cell immunity. But just a caveat is that these are in convalescent individuals that after they recover from this severe infection, they do have robust SARS-specific T cell response. Then people started to find out, well, this is a nice assay, but when I look at uninfected individuals or unexposed individuals, they also have these these SARS-specific CD4 T cells. So does it mean that other people that are not infected are also protected? The answer is maybe not that. It's that so there may be some cross-reactivity from the common cold coronavirus with SARS-CoV-2, but having these SARS-specific T cell phenotype doesn't really guarantee protective immunity. It's just likely reflecting that it could be a cross-reactivity with the other coronaviruses. But in those patients who are documented to have COVID-19, the good news is that the percentage or the number or frequency of SARS-specific CD4 T cells, if you calculate the frequency, they correlate very nicely with the antibody titer. Note that this is the spike receptor binding domain IgG. These are the antibodies that are most likely neutralizing. And this study also has neutralizing assays that not only it's about antibody titer, but it's also correlating with neutralization. So yes, CD4 memory, CD4 help is correlating with the antibody titer with a very nice correlation. So there's no dysynchrony. They're going in the same direction, which is great. Then Shane Crotty's group tried to ask, so why do some people, especially the elderly, have a more severe disease? Again, from all these analyses and looking at clinical data and other things altogether, they found that something matters called naive CD4 T cells. You're like, aren't we talking about specific CD4 T cells? Why do we now talk about naive CD4 T cells? So it's like this. Typically, we as humans have 10 to the 11th T cell repertoire, saying that we can recognize up to 10 to the 11 different kinds of antigen stimulation. They're just some naive cells sitting there. Once the antigen comes, such as SARS, then like a few of them or some of the clones will respond, proliferate, activate, and develop memory. And that's the pool we have. We have such a big repertoire to respond to very many different kinds of antigens. So it is proposed in this paper that when we get older, we will lose some of this repertoire. Some fail to be maintained. And during the first dip of of the CD4 T cell depletion early in infection, this may further dampen this. Some CD4 T cell repertoire may just be gone because of the CD4 depletion. So because we lack the big good repertoire to respond, and that may be related to why some older people may have severe COVID-19. So if you use single cell methods to look at what these SARS-specific CD4 T cells are doing, they're actually quite interesting and different. When you compare SARS-CoV-2 specific CD4 T cells with common cold coronavirus or flu, because if you look at these, the so-called like UMAP plot or these dimensional reduction plots in single cells, you see that, well, the cell type that SARS infection induced looks quite different from those that is seen in common cold coronavirus or flu. So turn out these are very interesting cells, such as T follicular helper cells you can find here, or you can find cytotoxic CD4 T cells. See, this is unique, and this is only seen in SARS, but not in common cold coronavirus and flu. So this is very interesting. In this paper, it seems that, well, they see during severe COVID-19, there are more cytotoxic CD4 T cells and fewer Tregs, maybe reflecting the overall activated state in this severe infection. But we can do a lot of functional analysis using the single cell method. So our lab is interested in what's happening during the early onset. So it's a preliminary study that we took samples from the Yale impact study that we look at the SARS-CoV-2 specific CD4 T cells. We found that these SARS-CoV-2 specific CD4 T cells do develop very early. After symptom onset can be as early as the first week. We're in the process of doing functional characterizations. Final questions. With all these that we learned from these publications, can we develop durable immune responses to COVID-19? And can we stop the cytokine storm and revert virus-induced immune dysfunction? Great news coming out this week from Shane Crotty's group that they follow up their SARS-CoV-2 PCR diagnosed confirmed patients, including mild, moderate, and severe COVID-19. 6 months later, right now, more than 90% of the individuals will have good antibody titer, IgG producing B cells are there, and SARS-specific memory CD4 T cells. Well, we still can't say that having a titer means totally protective from secondary infection, but at least this is promising that you have mild disease, severe disease, 90% of the individuals. You do see these memory components standing there ready to fight. And you can see this very nice immunology that over the course of infection, T effector memory cells, when the infection goes down and wanes, T effector memory decrease, but central memory, which is your memory that you keep in your lymphoid tissue to keep this immune memory, start to increase. So this is very exciting. And second, how do we treat the cytokine storm and immune dysfunction? So this is in the Emory by the Bossinger and Pardini group that they tested this in the macaque model. As Angela said, these macaque models are like mild to moderate disease. The monkeys don't really die, and it's not really recapitulate real human infections. And they infect SARS-CoV-2 and started this JAK inhibitor baricitinib 2 days after viral infection. This is almost impossible in human clinical trials because, if I get a COVID-19 today, I may have started coughing 3 days later, and it's probably going to be a few more days before I get any treatment in the hospital. So this is a very short period from virus to treatment. But anyways, as a proof of concept, they're testing if we block those JAK-STAT pathways downstream of interferon, can we stop this systemic inflammation and cytokine storm using this JAK inhibitor baricitinib? So by looking at the monkey lungs and other single cell profiling methods and then the blood cytokine levels, the overall finding is that by giving JAK inhibitor, you can decrease inflammatory cytokine chemokines. You reduce the number of macrophages and neutrophil like it's going to stuff out your lung and get respiratory failure. You can reduce activated CD4 T cells. The great thing is about this is that we had fear that JAK inhibitor is somewhat still an immune suppressant. We want to keep the good interferon signaling, but we want to suppress the cytokine storm. We want to maintain a good T cell response because we still want the memory. Preliminary data at least suggests that this baricitinib JAK inhibitor treatment does not impair SARS-specific CD4 T cell response and does not really impair the type 1 interferon signal. So that is great. So although it's still unknown whether this can be successful in humans, and I read it from Merkel Perding's Twitter, is that FDA already approved the emergency use of baricitinib and in combination with remdesivir, the antiviral drug, and as an emergency use for COVID-19. So in summary, I would like to thank my lab who maintained our productivity and integrity during this difficult time. And this is Jack and Risha who did our preliminary studies. We're still processing those samples. This is what they look like in normal times and in COVID times. We're very thankful to be at Yale with a very good like a clinical and basic science collaborating Yale impact study team, and Akiko Iwasaki's lab, her postdoc Carolina, and MD-PhD student John was provided just like tremendous help for us to like look at the system and get us involved in the BO3. All right, thank you. I'll just take questions.
Thank you, Dr. Ho, for that very in-depth presentation. So, as mentioned, it is now time for our Q&A session, and we do have several questions already submitted, so let's get right to them. First off, I'd like to ask Dr. Rasmussen if it's possible to kind of go over the models that she mentioned, and also the models that Dr. Ho also mentioned as well. Why are hamsters and macaques, are there any special properties that make them very good or at least popular COVID-19 models?
Yeah, that's an excellent question. So some of this has to do with the ability of those different model systems to accurately recapitulate human COVID-19 disease. And some of it is just really practical. So at the beginning of the pandemic, it was really apparent that we needed to develop animal models that could recapitulate COVID-19 in humans as quickly as possible, which is really challenging to do when it's a brand new virus that nobody's ever worked with before. And you don't really know what kind of responses you're going to get in a given host. So really what you do is you take the animals that you have in-house that you have experience working with, and you start infecting them, and you start to characterize those different models. So macaques are often one of the first models that are developed for any human disease, just because non-human primates are more similar to humans evolutionarily. And oftentimes the FDA will also require non-human primate data in order to approve any drugs or vaccines as part of their so-called two-animal rule. So often the first models that they develop are either rhesus or cynomolgus macaques. And in fact, that's what happened. And my collaborators who actually did the macaque studies at Rocky Mountain Labs, which is a NIAID facility in Hamilton, Montana, they have rhesus macaques that they can get, and they have a high containment lab that is already set up to do that type of non-human primate work and to do that chest x-ray, the imaging techniques that I mentioned briefly. So it was really a practical reason. You have the monkeys in house, you can get right to work. And so that's why that model was developed. But it turned out that in that model, while it's good for looking at mild or moderate disease, it's really not sufficient for causing severe disease. And very early on also, people tried to infect laboratory mice. That's an animal model that many people who do animal work have already in-house. But SARS coronavirus 2 doesn't efficiently bind mouse ACE2. So there was really no disease. So people used a transgenic human ACE2 mouse. And depending on the strain background of the mice, you get different levels of disease, but then you get the criticism that transgene expression isn't always the same as ACE2 expression in humans. So people do still use mouse models. Now there is a mouse adapted virus that has been developed by Ralph Barrick's lab. I think that people will probably start using in lab mice. So that has some utility. But I think really people also tried ferrets early on because ferrets are a great model for influenza. particularly for addressing questions related to transmission, because the respiratory tracts are more physiologically similar to ours. Ferrets will cough and sneeze, for example, whereas mice don't. So people also tried ferrets, but unfortunately, ferrets don't really get sick at all, even though they get infected. So that wasn't a good model for looking at severe COVID either. And that's when people start looking at some of these other models, such as the Syrian hamster, which does develop more severe disease. There are benefits and drawbacks to all of these different animal models. So really, you always want to make sure that you're using the right model to address the particular question you're asking. If you're asking a question about transmission, but you don't really care very much about the effect on pathogenesis, then a ferret model is perfectly fine for that purpose. If you want to know something, though, about what causes disease to become more severe, then you would want to use a different model that actually is susceptible to severe disease. So that's really some of the calculus that goes into choosing an animal model. I think going forward, there really is a place for all of these different animal models within COVID research, depending on what type of scientific question you're trying to address.
Thank you, Dr. Rasmussen. Next, for Dr. Ho, is there any relationship or correlation between T cell counts or proportions in the peripheral blood relative to their localized infiltrated numbers in the lung or upper or lower respiratory tracts?
That's a very good question. So, for example, when we see low CD4 counts in the blood, you see, oh, maybe they're just like being recruited to the lung, and that's certainly possible. Our data is limited. For example, in the single cell data in a bronchoalveolar lavage data set, that they see a large increase in CD8 T cells, but they didn't report a large proportion of CD4s. So we don't know whether CD4 are just lost or they're there. But bronchoalveolar lavage, it's a lavage, so it doesn't really puncture through the lung and pull it out and look at all the cells in there. It's just washing something from that for a quick sampling. So we actually don't know. I would say it's possible that they're attracted to the infection site, such as the lung. That's highly possible. But I believe that there are also the direct cytotoxic effects by TNF and then fast lichen, for example, that's already shown that they're causing these big drops.
Thank you, Dr. Ho. We have a lot of similar questions about persistent COVID, the so-called dubbed long runners. So I'd like Dr. Rasmussen to begin, and perhaps maybe just be kind of a generalized question. How does what we know about the immune response and immune overexpression and underexpression in various circumstances, how does that translate potentially to these long runners and these people who have COVID symptoms for months and possibly years?
That's an excellent question. And I would say that the short answer is that we don't really know. But a longer answer about that is that almost certainly the immune responses and the inflammatory responses are probably inextricably linked to the various presentations of so-called long COVID. I think that long COVID itself is something that we don't really have a good idea of the extent of it. I mean, there are a number of different types of presentations of that can be very different from one another. Everything from neurological symptoms, the so-called brain fog, to symptoms that sound an awful lot like a myalgic encephalomyelitis or chronic fatigue syndrome, to potential lung injury that is persistent. That might actually be caused by physical damage to the lungs, which we know as well that SARS coronavirus classic can induce, as well as MERS coronavirus. So it makes sense that SARS coronavirus 2 can probably also damage the lungs in that way. There have been heart problems reported. There have been vascular complications after people have recovered. So this is really a very protean condition that probably you can probably divide people into subgroups in terms of them having different mechanisms for what is causing, the umbrella of long COVID. I think that, one question that I have about it is how many of the symptoms are due to direct tissue damage caused by the virus or caused by the immunopathological responses to infection versus how many are caused by essentially a reprogramming of that person's inflammatory responses in a given tissue. In particular with the neurological effects of it. I think that, it's hard to say if the virus is actually killing cells in the brain or in the central nervous system. I mean, certainly there has been infection reported in neurons and nervous tissue, but it's very unclear to me if maybe that infection, rather than causing direct damage to those cells, is actually fundamentally changing the way that resident macrophages or resident immune cells are behaving within those tissues. And that effect lasts for a long period of time. So again, my short answer is I don't know, but I will be shocked if there is not a huge component in some kind of aberrant immune or inflammatory responses in the pathogenesis of long COVID.
I agree with what Angela said. I would say three things: the local structure, local immunity, and false positivity. So first, the local structure is that if you expect someone to have a very damaged lung, then there's a little bubble in there. It's just like afterwards, when the city is destroyed by bombs, it just takes time to really recover. So there could be some little niche here and there that the virus is not wiped out because the local destruction. And second is the immunity that's likely related to the underlying immune disease, such as diabetes or other like aging and others, that you need very robust antibody response and then inside the lung to really clear every single one of them out. And then you have CD8 T cells to kill all the infected cells. So every component needs to be very robust. So there may be some people who just don't have like robust immune responses, like in the very first like couple of weeks. And third is false positivity. Like maybe they are still real virus PCR detected, but they're dead viruses. They're somehow already like left over shedding from the dead cells that are not infectious. This is possible, but we don't know. But again, the short answer is we don't really know.
I think Dr. Ho, you touched on this very briefly just in the answer to that last question, but I'd also like to ask both of you, starting with Dr. Ho, do we have any data on COVID in immunocompromised individuals who would have weakened immune function and be more susceptible to infection, but also theoretically would have lower risk of cytokine storm?
That's a very good question. And for example, so this week, I think there is a paper coming out in CID, maybe looking at the SARS infection in HIV infected individuals. I haven't looked deeper into that paper yet, but this is something interesting. And there are other specific disease groups, but even in diabetes, for example, the immune status of diabetes patients is a range of the spectrum of immunity. So I would say the immune compromised status will affect viral control, but as what Synchronics data show, that majority of the patients, say 90% of the patients that they profile, 90% of them develop good memory T cell, B cell, and antibody titers. So I wouldn't say that 90% of all these people are all healthy. They must have some underlying, but they can still develop good immune responses.
Dr. Rasmussen?
Yeah, and I'll just add to that, that recently a couple papers have come out describing an individual patient. I believe both of them were either, they had a hematological malignancy. They had, I think, lymphoma in one case. And in another case, it was a patient who was undergoing treatment for cancer. Those are certainly both patients who would probably be immunocompromised or immunosuppressed to some degree. And there was persistent infection reported in these patients. Now, we don't have evidence that persistent infection is happening in everybody, but it certainly seems plausible to me that, as Dr. Ho was just saying, if a patient is immunocompromised, there may be issues with actually clearing the infection itself. So it does appear that being immunocompromised in some way may affect the ability to clear the virus. Whether or not that relates to disease severity over the course of their infection, it's really hard to say with the number that we've seen, the number of cases that have been reported. But it's certainly something to look into. In particular, I think looking into that with regard to HIV patients, where there is a huge population of HIV patients taking drugs that control their HIV, will have varying degrees of immune suppression. I think it's a really important question to ask, and that's in fact why HIV positive patients were also included in some of the vaccine phase 3 clinical trials, to make sure that those vaccines could perform adequately and provide protection in those patients that do have HIV at least. But immunocompromised, immunosuppressed is actually a very broad term that can refer to a variety of different conditions that may be different in terms of the mechanism of the immunosuppression. So it's really hard to generalize about this, but it's definitely an area that we need to take a closer look at.
Thank you both for those responses. I think we have time for one, let's not call it a question, let's call it a topic of discussion. And we have quite a few audience members wanting to know if there's any correlation or any relationship between symptom severity and the robustness of post-infection memory development. And I want to let Dr. Rasmussen answer first, please.
Yeah, so that's a really good question. And I, again, will say I don't really know the answer. But just in general, I think that robust immune response to the virus, in general, both innate and adaptive immune responses are associated with more rapid resolution and less severe disease. I think, though, disease severity, as Dr. Ho had spoken about in her talk, disease severity has been linked to some of these various depletions of certain types of T cells, the T follicular helper cells and the germinal centers that both of us talked about, things like that. But across the board, I'm not aware that we've found the smoking lymphocyte, to use that term, the smoking lymphocyte that proves that, if you have fewer of X cell or X cellular responses, you're going to have more severe COVID no matter what. I think it's a really complex question, and Dr. Ho did an excellent job of showing exactly how complex, especially the adaptive immune system is. So again, short answer, I don't know, but I think that's something that we do really need to look into, as it does have implications as well for things like biomarker development. How could we better triage patients who might need one treatment or another, or who might need to be hospitalized versus not? I think that it's a really important question to ask, and we need to be looking harder at predictors of disease severity.
Yes, I agree with what Angela said. I will add a few more comments. So first, there those high-dimensional analysis papers, say Akiko Iwasaki's paper and John Werrick's paper. Many of those papers take all these clinical parameters, including fever, underlying comorbidities, obesity, like hypertension, and many disease scores into consideration. You will see those many very colorful heat maps that they are also trying to figure out that what's the best marker to predict severity or immune response. So they all have those beautiful plots, but for what Dr. Akiko Iwasaki's paper showed is that, well, they name, say, interferon is one of the marker that is indicating disease severity. There are other cytokines that they named. But specifically for fever, for example, typically we think if I have a fever today, I would think this is the reflection of an interferon response. Either I get a rhinovirus or I get a COVID, anything. So it's an interferon response having fever. Yeah, that's sort of what I would think. But in COVID-19, it's messed up. See, the COVID immune response is complicated. And we know that COVID, you don't need to have fever. You can still get infected. It's too late and too little. So I may have a bad disease, but I don't have a fever. And second thing is that there are all these clinical observations that older people or immunocompromised people, they don't develop fever. For example, if I am autoimmune disease, I'm taking immunosuppressants such as long-term steroid, I may not have a fever, but it's just I don't have this fever, but I have a very bad disease going on. So it gets very complicated to say if fever is a predictor of memory response. It usually is, for example, in a classic animal model example, yes, but in COVID, it gets very complicated.
Related to that, and the answer might be we don't know again, but is there any correlation then between symptom presentation and immune time course, immune activation progression?
Well, I think Dr. Ho had a really nice figure in her paper from that recent review by Mujsevic and colleagues showing that, virus load peaks often before symptoms become completely apparent. And this is one of the reasons why the pandemic itself has been so difficult to control. But I'd be very curious to hear Dr. Ho's answer on this about timing of the various immune responses, because generally in my work for SARS-2 or any other emerging virus, a delay in the innate response does impact symptom severity. But I'm not sure that we have a great answer to, is this conclusively determining symptom severity in terms of, when X immune response goes up and the type of disease that a person will have.
Yeah, I agree. This is a complicated question, but usually symptoms should represent like viral load interferon. Again, this is disconnected in the sense of COVID. When we learned this from the example of HIV, for example, HIV may have like early peak of viral load, but if we treat patients very early before they really peak, you start treatment, that really restored all the immune responses. So I would take it into a different answer is that if we have like SARS symptom onset, that if we give some early treatment, say really early remdesivir or early kind of antivirals, just like you take Tamiflu in the first 24 hours of influenza, or you take acyclovir in the first 24 hours of herpes zoster, that they can probably change the disease course and ameliorate symptom. But like things after 3-5 days is getting complicated between the battle of in the immune system and the virus. And there's a question in the chat saying that why don't we use like interferon alpha spray in SARS-CoV-2? I actually don't know, because that was provided in MERS infections for healthcare workers. They just have a interferon alpha like nasal spray to prevent. But I don't really know the administrative part that why we don't try this in SARS-CoV-2.
So Dr. Ho, I can actually answer that. I think
Sure.
that those trials are going on right now.
Sure.
I think the issue is that, and this is just what I've heard from Ben Tenover, I believe, that you actually can't use interferon as a true prophylactic just because repeated administration intranasally causes like the mucous membranes to dry out. It basically, it messes up barrier function, and I guess it's just pretty uncomfortable. But I don't really know, and I think it is in a clinical trial right now. I don't really know why they're not treating patients at the time of diagnosis, especially people who are asymptomatic with interferon intranasally, or potentially, intranasally would be the way to do it, I guess, or nebulized. But I think that those studies are ongoing. I just don't think we have an answer. And I think that some of it has been a little bit, people have lost their enthusiasm because of the results of the Solidarity trial, which was using interferon in hospitalized patients. So that I completely agree that it's going to be less likely to work in those patients, just like remdesivir is, and just like monoclonal antibodies are, because as you pointed out, that by that point, you're not really fighting against the virus, you're fighting against the immune response or the dysregulated inflammation.
Sure, I totally agree. I think what we know about interferon is that timing is so important. So you cannot give it 7 to 10 days. If you give it, that may be the culprits leading to severe disease. So timing is so tricky.
Thank you very much to both of our speakers today. Unfortunately, that is all the time that we have. If you have any further questions, please consider reaching out to the speakers directly, and their emails are presented on the screen. As a reminder, this webinar will be archived on the scientist website, and you will receive an email notifying you when the on-demand webinar becomes available. I would like to thank everyone who took the time to join us today, especially those of you who shared your questions and your comments. And on behalf of the scientist, I'd like to extend a heartfelt thanks to our speakers, Dr. Angela Rasmussen and Dr. Ya-Chi Ho, as well as our sponsors, Sinobiological, 10x Genomics, Selecta, BPS Bioscience, and Canopy Biosciences. Thank you, everyone, and have a pleasant day.